Hi, many thanks for inviting me to talk about the Orchestra project, which is looking at the foraging ecology of common guillemots and razorbills in the non-breeding season and the potential consequences of displacement from offshore wind farms for these species during this part of the annual cycle. Research on the impacts of offshore wind farms on seabirds has so far been focused mainly on the breeding season. In contrast, offshore wind farm effects in the non-breeding season, when the birds also face considerable energetic and demographic challenges, have received much less attention and were identified as a priority evidence gap by ScotMER. For rock species such as the guillemot and razorbill, the post-breeding molt and mid to late winter are periods when the birds experience high energetic demands and form large aggregations offshore, and so may be particularly vulnerable to anthropogenic effects. So it's important for planning and impact assessment purposes to better understand the potential effects of displacement from wind farms at these times, which may alter foraging behavior, energetics, distribution, and ultimately survival rates. And several recent studies have increased our understanding of these key aspects of wintering ecology. And the geolocation data from the multi-colony study by Buckingham et al. Were used to carry out the first winter apportioning of birds of known provenance. And despite these recent advancements, several knowledge gaps remain. For example, to do with interannual variation in distributions, behavior and energetics throughout the non-breeding season, and in particular winter diet. So in Orchestra, we are hoping to address these evidence gaps, and accordingly, the aims of the project are to firstly quantify the distribution, foraging behavior, and energetics of guillemots and razorbills during the post-breeding season and in winter. So these are the key periods of vulnerability that I mentioned earlier. Secondly, to obtain information on non-breeding season diet via stable isotope analysis of feathers and the bird's potential fish prey. Thirdly, to assess the extent of variation in foraging strategies and prey during the non-breeding season, the energetic consequences of this, and implications for offshore wind farm displacement effects. And finally, towards the end of the project, to incorporate the findings into impact assessment tools to help inform marine policy and planning. So how are we planning to do all this? The data collected in 2024 to 26 at 8 guillemot and razorbill colonies in Scotland, and I'll show you a map of these at the next slide. We deploy two models of integrated GLS TDR tags on leg rings, both of which record light levels, pressure, saltwater immersion, and temperature at high resolution. And you can see how they look here. They record data over the whole non-breeding period and are then retrieved in the following year. And since we have not used these particular tags previously, we also do monitoring for device effects. We also collect feather samples that are so basically from feathers which are formed during the post-breeding period and in midwinter to infer diet during these key periods. And we additionally collect samples for sexing. And finally, we use fish samples collected during Scottish Government bottom trawl and sand eel surveys from areas overlapping with the non-breeding distributions of our study colonies, and the survey areas are shown on the maps here. And we do this to obtain isotopic signature from the likely prey of our birds. So here we have a map of the colonies included in the study, and as you can see, they cover most of the breeding range of the two species in Scotland, and were selected also because they are predicted to interact with wind farms in the non-breeding season. The table shows the target sample sizes for tag deployments at each colony, which are based on our previous experience of availability of birds and retrieval rates at these sites. So how did the first year of data collection go? Well, the 2024 field season unfortunately was very challenging with poor return rates and delayed breeding or non-breeding at a lot of the colonies. The photos here show the same study plot on the Isle of Man in 2023 and 24, and as you can see, the density of breeding guillemots is much lower in 24. Despite this, we managed to deploy 72% of the planned tags on guillemots and 66% on razorbills and collect feathers from all tagged individuals. And the target sample sizes were achieved at all West Coast and some of the East Coast colonies, but the biggest challenges were faced in the North and Northeastern colonies, where much fewer or no tags were deployed. And the collection of fish samples went as planned, and we have plenty to use for the upcoming stable isotope analysis. And at the end, I'll give you a very brief overview of the analysis we are planning in order to address the project aims, but I hope that we can present more in more detail on these at future ScotMER symposiums once we have results. Firstly, we'll be looking at space use, and for that we'll produce monthly utilization distributions with uncertainty for our tracked individuals, and then aggregate these to derive colony level estimates of spatial distribution. And as part of this work, we're also reviewing methods for analysis of geolocation data and exploring potential for methodological improvements. We'll also quantify foraging depth and time spent diving at daily scale throughout the no-breeding season, and combine these foraging metrics with utilization distributions and population sizes to estimate time spent foraging within a defined space at a defined time. We'll estimate non-breeding season diet and look at variation in diet between individuals, colonies, and over time. Next, we'll derive daily time activity budgets and energy expenditure for each individual throughout the non-breeding season, and then combine these estimates with data on space use and to quantify spatial variation in time energy budgets. And the final goal is to incorporate all this information into impact assessment tools. This will involve producing improved estimates of non-breeding season apportioning and also estimating the consequences of displacement from offshore wind farms. And to do this, we'll treat areas in and around the wind farms as zones of varying severity of habitat loss, leading to a redistribution of birds into surrounding habitat. And we'll then estimate the consequences of this redistribution for time energy budgets to understand when and where the consequences of displacement may be most acute. And the findings would then be incorporated into existing impact assessment tools like the cumulative effects framework. So thank you very much for listening, and apologies for not being able to join the Q&A session, but Francis will be there to answer any questions. Thank you.
Good morning, everyone. My name is Claudia Tapia Harris, and I'm a conservation scientist at the RSPB. Today, I'm going to talk about the RSPB-led project titled Bird Responses to Avian Influenza and Developments at Sea, or BRAIDS for short. So here on the slide, you can see a short list of names of RSPB colleagues that are currently working on the project. However, BRAIDS is a massive collaborative effort involving many individuals from different organizations, mostly across Scotland, and with the possibility of bringing in additional collaborators in the future. So as well as having a large number of collaborators, there are also several funders involved in the project. The principal funder is the Scottish Government through the ScotMER programme, but others such as the Fourth and Take Consented Developments, as overseen by FATRAG, the ScotWind Constrained Developers from East and Northeast Leasing Zones, and DEFRA, as administered by Hartley Anderson, are also important funders. So the main aim of BRAIDS is to address two of the major current threats to seabirds, the rapid expansion of offshore wind developments, and the ongoing H5N1 highly pathogenic avian influenza outbreak, which from now on I'll be referring to as HPAI. Our main goals are to track seabirds to determine their distribution and behavior during the different phases of offshore wind farm construction, and to study the extent to which seabird foraging ranges and behavior may have changed as a result of HPAI-induced reductions in colony size. So we have 4 different target species. They are the black-legged kittiwake, northern gannet, common guillemot, and razorbill, and all of which have been identified as being highly susceptible to both of these threats. So the project will generate as well as use different type of data, such as current and historical seabird tracking data, blood samples and cloacal and clonal swabs data, as well as census and population counts, and prey data obtained from projects like Prepared. But here I'll focus on the first three of these. So one of the biggest components of braids is to deploy GPS tags on the 4 different target species across multiple colonies. As you can see from the map, most of these colonies are located on the east coast of Scotland. The stars represent the sites that have been approved for tagging, and their color represents who the lead organization in charge of the tracking there is. So the bird icons represent the study species that will be targeted at each site. So going through this very quickly, the RSPB will be leading kittiwake and auk tracking at Coppensay, Pentland Firth Islands, and Falshew. We'll also be doing a bit of kittiwake tracking at St. Abbs Head, jointly with the University of Glasgow and Aarhus University. We'll be tracking gannets both at the Bass Rock and Elsa Craig. The BTO will be leading kittiwake and auk tracking efforts at the Buchanest to Collieston Coast SPA, and UKCEH will be leading tracking efforts at the Isle of May. As you can see, there are two additional sites that are shown with a dot and in italics, so Troop Head for gannets and Dunbar for kittiwake. And these are additional sites where we haven't done any tracking yet, but we are looking into to see whether it will be feasible to do so for 2025 and 2026. But confirmation on this is still pending. So without going into much detail, these tables show the sample sizes for each species of the GPS tags that we have or that we want to deploy at each site over the course of the 3 years, so from 2024 to 2026. And the main idea here is just to show that we aim to deploy around 560 tags on kittiwake, 290 tags on auk, so including both guillemot and razorbill, and over 230 on gannet. So which, if all goes well, will give us a massive joint effort of over, hopefully, 1,000 tags deployed over the course of the life of the project. So braids will also rely on already available data. Most of this data has already been collected by collaborators themselves. Here is just an example of 3 maps of tracks collected through the FAME and STAR project, which ran from 2010 to 2015. So this is just to show that there's quite a bit of available tracking data for our target species at our target study sites as well. And similar goes to gannets at the Bass Rock. Most of the tracking at this site has been led by Leeds University. So by using historical as well as current seabird tracking data, we aim to monitor changes in seabird distribution and behavior pre and post wind farms, as well as during the course of the HPAI outbreak. So another important aspect of braids is to collect blood samples and cloacal and clonal swabs of individuals of all age groups of gannets in order to detect the presence and or absence of live HPAI virus. And this will be done in collaboration with the University of Edinburgh, as well as the Animal and Plant Health Agency, APHA, as part of the EcoFlu and iPrepare projects. So this last slide is to show the different analytical packages. These are shown in blue, and to the left you can see what organization will be leading on these analytical packages. And I guess the main idea of this slide is just to show that results from this project in terms of understanding the scale of offshore impacts in the context of HPAI on seabirds will be of direct relevance to impact assessments. So braids or projects like braids bridge the critical evidence gap between scientific research and practical applications for use in marine development assessments. So thank you to all of our collaborators, funders, project steering group members, and as well as you for listening. Thank you very much.
Hi, my name's Kirsty Franklin and I'm a Senior Conservation Scientist at the RSPB. And today I'm giving a really brief overview of two storm petrel tracking projects that the RSPB are involved in. The first is a project commissioned and funded by the Scottish Government Marine Directorate, which I'll refer to as the West Coast Petrel Tracking Project. And the second is called Prosolariform Behaviour and Demographics, or PROC-B, which is part of the Offshore Wind Evidence and Change Programme led by the Crown Estate. For PROC-B, I'll just be focusing on the RSPB-led component, focused on storm petrel tracking, but you can find out more about the project as a whole by listening to Bryony Baker's talk on Friday. So what are these projects all about? The West Coast Petrels project is focused on improving our understanding of the marine distribution and behaviour of two petrel species, the Leach's petrel and the European storm petrel. Prior to this project, there had been no tracking at all of Leach's storm petrels in the UK and only one study for European storm petrels, but in Shetland. So for this, we undertook tracking for Leach's at their largest UK colony on St Kilda and for European storm petrels on the Cheshire Shoals. Both projects are also looking at the impacts of offshore wind farms on storm petrels in the UK. For PROC-B, this is focused around developments in the Celtic and Irish seas, hence GPS tagging of birds at Lundy and the Isles of Scilly, as well as collecting data on storm petrel flight heights for use in collision risk modelling. And for this, we focus on the largest colonies for each species in the UK, so again on St Kilda and then Musa for European storm petrels. Both projects have a common theme in that we're collecting bird-borne tracking data by attaching tiny tags to individual birds. The small size of these seabirds means that the tags must be archival. That means we have to catch the tagged birds a second time to retrieve the tags and obtain the tracking data, really adding to the challenging nature of working on these small nocturnal and secretive birds. For both projects, we're using conventional GPS tags to collect spatial data, whereas for flight height, last year we trialled two small devices, a high-resolution GPS logger, and a second tag that has no GPS functionality but collects continuous pressure and accelerometry data. Into some results. So for the West Coast Petrels project, tracking was conducted in 21, 23 and 24, with delays and gaps in tracking due to COVID and HPAI. But across these years, we achieved really high retrieval rates, collecting information on a good number of individuals. It's clear from these plots that these birds are deep water specialists, consistently travelling northwest of the colony and often over really long distances. You can see in 2024, one bird making it 2/3 of the way to Iceland. However, although several offshore wind development areas are within the foraging radius of these birds, none of the tracked birds overlap with the wind farm areas, so that's good news for the Leach's storm petrels. Again, for European storm petrels from Tresnish, we retrieved a really good number of tags. The low number of tracked individuals in 2024 was just due to a delayed breeding season, but you can clearly see some patterns across these maps. The European storm petrels generally tend to forage much closer to the colony than the Leach's storm petrels, and also over much shallower waters of the continental shelf, rarely venturing beyond the shelf edge. In 21 and 24, birds were mainly concentrated to the west of the colony, whereas in 23, which we know is a particularly bad year for the storm petrels, birds travelled much further and in a greater range of distances. Across all years, quite a few of the tracked birds entered one of the wind farm development areas directly south of the colony, and in 2023, one bird actually ventured into multiple lease areas off the coast of Ireland, really showing the importance of multi-year tracking and across different environmental conditions. To continue with the spatial data we've collected, I'm now taking you to the southwest of England as part of the PROC-B project. We've only conducted 1 year of data collection in 2024, and this is the first attempt of tracking European storm petrels from English colonies. The highly challenging nature of this work is reflected in the fact that we only managed to track one individual per colony, but even still, we've collected some really fascinating data. In light pink, we have the Isles of Scilly bird. We tracked this individual over one foraging trip, but a really long one, over 1,300 kilometres, all the way to the Celtic Sea continental shelf. And this bird's actually behaving a bit more like our St Kilda Leach's petrels. Whereas the Lundy bird in dark pink, which we tracked over multiple foraging trips, stayed much closer to the colony, behaving more how we would expect from European storm petrel. You can also see that only the Lundy bird overlapped with any of the proposed lease areas. But obviously, these data are just from one individual from each colony, so we do not yet know to what extent these tracks are representative of the colonies as a whole, and more tracking data is needed. Now on to flight heights. First, I'm just showing you the raw data from the high-resolution GPS tags, leeches, storm petrels on the top and Europeans on the bottom. Here you can see these tags take bursts of GPS data every 2 hours, and within each burst we have 1 second GPS data, which is why these look like beads on a string. Associated with each GPS fix, we then have a GPS-derived flight height. So in these last plots, you can see the raw flight height data represented as height above the ellipsoid. So just ignore the actual values for now, but the range in heights across these few minutes is quite clear. For these random individuals, once you exclude the first 30 seconds of data when the tag is still connecting to the satellites, the range in leeches heights is about 9 meters, and for the European storm petrels is only about 3 meters. We've only just started digging into this data, but currently it's looking really promising. Finally, I just want to show you some of the barometric pressure data from 2 random tags, again showing raw uncalibrated height data with leeches on the top and Europeans on the bottom. The plot for the European storm petrels just shows the individual to be fluctuating around a relatively low height, whereas the pattern for the leeches is really interesting. Because we have tagged these birds at the nest, we know exactly where the nests are and so how high they are above sea level. So despite not having locational data, we can tell that at this time, the bird was returning to the colony, and we can see when it started ascending to the colony into its nest at about 250 meters above sea level. So again, we've got a lot of data crunching to do, including calibrating these tags with known heights at known times. But so far, we're really excited by the data we've collected in the first year of this project, and we look forward to collecting more. So yeah, that was a really quick whistle-stop tour of our 2 petrel tracking projects. And I'd like to end it by acknowledging the funding sources for this project, and I'm really happy to take any questions. Thank you.
Hello, this is a brief overview of the now completed project INTAS, which stands for Integration of Tracking and At-Sea Survey Data. The problem we tried to tackle with this project can be illustrated with this cartoon here. Imagine that you have a survey area represented by this polygon, and that within the survey area you've collected some data which indicate certain densities and abundances of seabirds. The question is, what happens if you're especially interested in a particular class of bird, such as breeding birds, coming from a particular set of colonies or individual colony? How do you tease apart how much of the usage in this area, which may be a proposed region for offshore development, comes from breeders of a particular colony, and how much of the usage comes from other colonies or other types of birds, such as juveniles and so on? Now, intuitively, if you wanted to get an answer to this question, you would need to think about something like coloring the usage emanating from that colony, some form of tracing or tracking the birds that emanate from that colony. And the typical ideal type of data that can achieve that objective is, of course, telemetry tracking. So we have a situation here where we're interested in answering a question pertaining to survey data using telemetry data. And intuitively, those 2 data can complement each other so that we can address this key question of apportioning usage to individual colonies and breeding classes. So the task of this project was, first of all, to come up with a formal statistical method of integrating survey and telemetry data so that we could analyze their distribution from these 2 different types of information jointly as a function of environmental covariance. So essentially, a species distribution model, but one which relies on 2 very, very different types of data. And the second task was to try and apportion the effects of potential disturbance in particular contours or polygons to individual colonies and individual types of bird. I'm not going to go too much into or for that matter at all, given the time constraints, into the modeling approach for this, but you can find all the details of how that integration was achieved in a very recent paper we put out this year called Joint Inference for Telemetry and Spatial Survey Data. Essentially, what this paper does is to bring together two very well established approaches to spatial analysis. One you may be familiar as habitat selection functions or resource selection functions, primarily developed for survey data, and the second one called step selection functions, which is primarily developed for sequential data such as those that might come from satellite tracking. So those two sources of information were combined theoretically for the purposes of this project, and the details can be found in this paper. Very keen to get any feedback from you on that if you happen to read it. For the purposes of INTAS, we chose 4 relatively data-rich species and collated survey data from different impact assessment surveys carried out in the past, as well as telemetry data for each species collected individually for juveniles and breeders. The sort of cutting to the bottom line, I guess, the habitat modeling allowed us to look at commonalities in how the different bird species responded to different environmental covariates. So, for example, a preference for deeper water or versus a strong preference for deeper water is illustrated by the upward pointing green arrows here. We found that there were actually fairly congruent approaches by different species for some of these covariates. An interesting result. These are the sort of things you could probably get out of analyzing habitat preferences either using telemetry or using survey data in isolation. But the sort of key results of this technique were that it was able to return estimated distributions by separating out the effects of adults and juveniles from the survey data by allowing the information provided by the telemetry data. And more to the point, it was able to return individual percentages contributed to the usage of any one polygon at sea. Here's an example in this picture here, apportioned by colony and breeding class. So, for example, on average, 14% of the usage contained in this contour is provided by juveniles of this colony, as opposed to 8% provided by adults, with credible intervals accompanying these estimates. It's possible to return those in absolute terms if necessary, so that we can get an idea of what proportion of the total usage or by species is provided within a particular contour. So that's the apportioning job. And finally, mention on next steps. There are 2 key developments that we're pursuing with this framework into the future. The first one is of practical value. We're trying to incorporate this approach into the cumulative effects framework so that layers of species distribution by colony and breeding class can inform decision making. And methodologically, we're working actively to implement these sort of joint likelihood approaches within sophisticated and widely used frameworks such as in LaBrue, Bayesian methodology that allows for spatially explicit modeling, so that we can popularize and put these methods at the hands of analysts outside of our teams across the board. Thanks very much for watching.